Rachael Muscatello is a research assistant professor in the Department of Psychiatry and Behavioral Sciences at Vanderbilt University Medical Center. Dr. Muscatello has published extensively on the relationships between stress, development, anxiety, depression, and social functioning in autistic youth to better understand the role of physiological function in psychosocial behavior. These webinars are made possible thanks to generous donor support. If you'd like to contribute, please visit our website at autism.org. And now I'll turn this over to Dr. Muscatello.
Thank you so much, Denise. And thank you everyone for being here today. I'm excited to share some of our recent work regarding physiology and psychosocial functioning in autism, particularly examining the unique role of the autonomic nervous system. Oh, sorry. There we go. Before I start, I do want to acknowledge research support, particularly for the studies that I'll be talking about today. We've been very fortunate to have support from the National Institute of Mental Health, Autism Speaks, as well as through internal grants and core support. I do not have any competing interests to declare. Some of the data presented is unpublished or undergoing peer review, and so just please, the information should not be cited without prior consent from myself. Okay, so the goal of today is to hopefully address three kind of primary areas. So first, I want to define the autonomic nervous system and its role in autism spectrum disorder. And then to describe how the autonomic nervous system and stress and physiological functioning may also relate to social behavior in autism. And then finally discuss potential clinical implications and critical avenues for future research. So first, defining the autonomic nervous system, or ANS. The autonomic nervous system is divided into two branches. The first is the sympathetic, or SNS, which is often termed our fight or flight branch. So we think of this as responding to a stressor with increased heart rate, raising blood pressure, increasing respiration, among other things. Whereas the parasympathetic, or PNS, rest and digest branch is more about recovering from these stressful moments with slowing heart rate and lowering blood pressure. And in the image in the center of the slide, we can see that ultimately both branches of the autonomic nervous system innervate a number of organ systems, just highlighting the ANS's influential role in the body. And again, generally speaking, when we think about an increase in sympathetic activation, we think of that as an increase in arousal or stress, whereas an increase in parasympathetic function would generally be thought of as a decrease in arousal or a decrease in stress. So there are a number of ways that we can measure autonomic function, but one way in particular that is very beneficial is heart rate variability. This is a non-invasive measure that uses electrocardiography, or ECG, to detect changes in the beat-to-beat variability of heart rate. And it's particularly useful as it can provide insight into the contributions of both the parasympathetic and sympathetic branches. One of the metrics that we use extensively in our research is respiratory sinus arrhythmia, or RSA. This measure is a cardiac measure of parasympathetic regulation specifically, where elevated RSA or higher RSA indicates more parasympathetic regulation. Another metric that we use is pre-ejection period, or PEP, which is an indicator of sympathetic cardiac control. And this measure is actually a time-based measure of the amount of time between the electrical stimulation of the heart and when it actually pumps out blood. And so with this measure, when we think of lower pre-ejection period, so a shorter time between electrical stimulation to pumping, that actually indicates an increase in sympathetic activation. And I'll bring that back up when looking at some of our results, because sometimes it's can be difficult to remember and seems counterintuitive at times. So why do we care about the autonomic nervous system in the context of social functioning? Well, it's a pretty long answer, but essentially there are a number of theories and models that have been developed, such as the neurovisceral integration model, thinking about the central autonomic network, or the polyvagal theory is just some of the examples. And ultimately, what all of these theories highlight is that there are a number of regions within the brain that are responsible for many processes, such as executive attention or emotion regulation, but these regions are also involved in regulating the autonomic nervous system. And so this overlap in brain regions might have important implications not only for physiology, but also psychology. And so the polyvagal theory, as one example particularly relevant within autism, is this idea that these calm autonomic states, so in particular, periods of higher parasympathetic regulation, actually promote social behaviors that are interconnected through what's termed the social engagement system. And this is seen in several studies that have, in fact, reported that higher parasympathetic regulation is associated with more sociability, improved social communication, just as some examples. And that leads us to this idea that given the hypothesized connection with autonomic arousal and social engagement, it may be particularly useful when we think about the autonomic system in the context of autism spectrum disorder. And as I think most everyone is probably very familiar with at this point, but just to remind ourselves of the DSM-5 criteria for autism, there are two primary diagnostic criteria, including persistent deficits in social communication and interaction, as well as restricted and repetitive patterns of behavior, interests, or activities. And for the purpose of today's talk, we're really focusing in on this first one of persistent deficits in social communication and interaction. So first, talking about this link between autism and physiology specifically. We have published a number of studies now that have looked specifically at the autonomic system in autism. And we find in several of our studies reduced parasympathetic regulation in adolescents with autism. So when we're looking at this graph on the right, we have respiratory sinus arrhythmia, or RSA, that measure of parasympathetic function on the y-axis. And then in red, we have our neurotypical group, and in blue, our autistic group. And what we see is that overall, throughout the entire period that we're measuring, the neurotypical group has significantly higher parasympathetic regulation. But one of the things that we've really been highlighting in the last several years is the fact that there are other variables that we need to consider when we think about whether or not there are differences in autonomic function within autism. And one of those variables that we have looked at quite a bit is body mass index, or BMI. And this is because autonomic functioning and BMI are highly correlated. And in fact, in one study of adults, weight loss was actually associated with an increase in parasympathetic control and reduced sympathetic activation. And you might recall that again, that parasympathetic is our kind of rest and relax, whereas sympathetic is our fight or flight. And so in general, having less of the fight or flight active and more of that rest and relax would be beneficial. Additionally, there are high rates of overweight and obesity in autism, including, we've cited this in a 2021 study with Dr. Blythe Corbett. And in that study, we found that around 40% of our autistic sample was falling into the overweight or obese category. And indeed, when we look at BMI specifically as it relates to RSA, we do see that when we're looking at the lower percentiles, which are in these blue colors, relative to higher percentiles when we start to approach overweight and obese, in the red, we do see that lower overall RSA or less parasympathetic regulation in those instances of elevated BMI. Additionally, just as of last week, actually, we published looking at these relationships longitudinally. So looking at them over a 4-year developmental window. So in this study, we were focused not only on BMI, we were also looking at medication usage, as well as just change over development, particularly the adolescent time period. And the first thing that we noted was that although the neurotypical or TD, typically developing youth in blue, had somewhat higher RSA compared to our autistic youth in red. The overall RSA remained stable over adolescence for both groups. There was not a difference in that developmental trajectory. Additionally, it's not shown in this graph, but the diagnostic differences were largely driven by BMI as well as medication usage, particularly things like antidepressants. In contrast, the sympathetic branch, we did not see diagnostic differences either in overall pre-ejection period or over development. So again, as a reminder, pre-ejection period, higher PEP is actually an indication of less sympathetic control or activation. And so for both groups, regardless of diagnostic status, there was a steady decrease in sympathetic activation over advancing age and puberty. So all of that to highlight, first of all, when we think about our physiological differences specifically within autism. So although we have noted some diagnostic differences in parasympathetic regulation, as of now, we have not seen any evidence for differences in sympathetic. However, as I'll talk a little bit more about later, we do still need to dive into that area even more and really focus on that sympathetic branch. Importantly, these diagnostic differences, though, may largely be driven by some of these other variables that we need to consider, like BMI and psychotropic medication usage. In our studies in particular, we've definitely seen that where it is those other variables that are driving these group differences rather than the diagnostic group itself. Both autistic and neurotypical youth do show similar developmental trajectories, which is encouraging to see, with stable parasympathetic regulation and reduced sympathetic control over time. And again, just getting back to this idea of a trait-based approach, it may be beneficial moving forward when we think about autonomic differences in autism to think less from a categorical autism versus not approach, but thinking more about what traits or individual subtypes might be related specifically to some of these more notable physiological differences. And then that leads us into the second part of this kind of triangle or relationship that we'll talk about, and to now include social interactions or social behavior. And this is relevant as social interactions may be a significant source of anxiety or stress for some autistic individuals. And the psychological stress overall, we can think of as a time when our environment is perceived as too demanding and based off the context or an individual's experience. And so that leads us here to now consider the link with social functioning or behavior or cognition within autism, but not only within autism, but how do they relate to each other, physiology and social functioning as well. So to measure some of these, or to look at some of these questions, we have utilized two primary paradigms that I just want to briefly review. The first is called the Trier Social Stress Test Friendly. It's based off of an adaptation from the original Trier Social Stress Test, which I'll describe momentarily. But this particular version is indeed just thought of as a friendly social interaction. Two children or peers are interacting with one another, are told they can just spend this time to share more about themselves. And so hopefully we would think that it would largely not be a stressful situation, just interacting with another person. In contrast, the original Trier Social Stress Test, or TSST, is more of a social evaluation paradigm that is quite stressful for many individuals, where they are tasked with giving a speech in front of two raters who are not providing any sort of feedback, no smiling, they just remain neutral. And the participant is told that their speech will be compared to others in the study to kind of include that evaluation component. So they have to give this speech as well as doing a serial subtraction task. So as we can imagine, this is often quite stressful for many individuals. So in terms of what we've seen so far in our work, so first focusing on that social interaction or the Trier friendly. Looking specifically at social behavior, we do note that our children and adolescents with autism engage in less verbal interaction and generally less eye contact. So in our bar graph here, we have the amount of time or the duration spent engaging in these different behaviors. And then we have on our x-axis, the first two are well-modulated eye contact and verbal bout or just interacting. And then the other two are poorly modulated eye contact or time spent silent. And our youth with autism in red do show less eye contact and verbal interaction relative to neurotypical peers, but on the flip side, more time with poorly modulated eye contact or an increased time being silent. As far as the physiological piece during these interactions, so this graph is the same graph that I showed earlier when I was highlighting the overall mean difference in RSA. But now I want to highlight the x-axis here where we're looking at time point. And so this is for the friendly. So we have a preparation period, the first half of the interaction, the second half of the interaction, and then our recovery. And so, as I had mentioned before, although the autistic youth in blue do have lower RSA overall, we actually don't see any reactivity differences. So these changes in RSA or parasympathetic regulation from one time point to the next do not significantly differ from neurotypical youth. We see a similar trend in looking at social evaluation procedures. So now the Trier social stress test, where once again, our autistic youth in red do have lower overall RSA compared to neurotypical youth, but that change from one time point to the next does not significantly differ. So the actual stress reactivity, at least within the parasympathetic branch, because this is all looking at RSA, does not differ between the diagnostic groups. What is interesting, however, and ties into that relationship between autonomic function and social behavior is that we find that regardless of diagnostic status in both the neurotypical and autistic youth, those who have higher social symptoms as reported on a parent report measure called the CBCL, those individuals also have lower RSA or lower parasympathetic regulation. So again, pointing to that idea that the increase in parasympathetic activation or activity is also helpful for promoting social behavior. Similarly, in another study of ours looking at a different parent report measure of social functioning, this time using the SRS, we find similar trends where those who had less or fewer parent-reported social difficulties had higher overall RSA compared to those with more parent-reported social challenges. But once again, what is interesting is that even though there is a difference in the overall parasympathetic regulation, there was not any sort of difference in terms of the reactivity or the change from one point to the next. And so with that, we so far have found that our autistic youth demonstrate similar parasympathetic reactivity, so that change over time when responding to different social stressors, regardless of the context. So whether it's a friendly interaction or a more evaluative, performative type stressor, the autistic youth did not differ in their actual stress reactivity patterns, specifically within the parasympathetic branch of the autonomic nervous system. However, regardless of diagnostic status, there was a relationship between an increased severity of parent-reported social symptoms and lower overall RSA. But it's important to note that up until this point, all of the research that I presented was specifically focused on the parasympathetic system. And so we do need to investigate further the extent to which sympathetic activation may differ. And this is especially important when we think about how sympathetic fight or flight is kind of what we think about when we think about responding to stress. And so does that activation change in these different social contexts will be important to look into further. So our final piece of the triad is the clinical implications, and particularly physiology and internalizing symptoms, so depression and anxiety, among others, but depression and anxiety are the two that we focus most heavily on. And I probably don't need to make the argument to this audience, but still just pointing out that anxiety and depression are very prevalent amongst our youth, with estimates as high as 20% of youth ages 12 to 17 reporting at least one major depressive episode in 2021, and as many as 32% with an anxiety disorder. But even more striking is within autism, those numbers are even higher. So even as young as 4 to 8 years of age, as many as 5% of autistic children in that age range are reporting depressive diagnoses, again, getting up to 20% into adolescent, and as high as 40% for a lifetime prevalence. So any history of a depressive episode by adulthood. And then as many as 1 in 2 individuals with autism may have an anxiety disorder. So these are two very important things to consider when we think about our clinical implications, and also just in terms of what we might need to be focusing on within this population. And so that leads us to our third piece of the triad with internalizing symptoms, not only in how they are specifically related to autism, but then also how do they relate both to social functioning and physiology. And so I just wanted to revisit some of these models and theories again to point out that, to reemphasize this overlap in these brain regions, where some of the same regions that are ultimately responsible for driving autonomic response or controlling autonomic response also overlap or are also involved in things like executive attention and emotion regulation. And so that overlap is important to think about when we're thinking about how physiology may be playing a role in our psychology or in our emotion processing. And in fact, there is quite a bit of data across a number of populations citing lower resting state parasympathetic regulation in depression, anxiety, as well as conduct disorders, non-suicidal self-injury, and many others that I haven't listed here, all to very much highlight that that autonomic function, parasympathetic in particular, might be a very important marker in terms of thinking about as a marker of psychopathology. In our own research, looking at the relationship with behavior and internalizing symptoms, both in autistic but also in neurotypical youth, we do find a number of important relationships here. The table here, there's a lot of numbers, a lot going on, but essentially what we see at the top of the table is a number of different behaviors, again, that verbal interaction and eye contact, as well as overall involvement and the quality of rapport in the interaction. And then on the left-hand side, we have both parent-reported and self-reported anxiety and depressive symptoms. And what we find is that for many of these behaviors, especially interaction or verbal bout, as well as involvement and rapport, those with higher depressive symptoms or higher anxiety symptoms show less time spent verbally interacting and have less overall involvement or poorer rapport with their peers. And in terms of physiology, we have looked at specifically at depressive symptoms or affective symptoms. And what we found is that in autistic individuals specifically, there is a really unique relationship where Respiratory sinus arrhythmia, or RSA, parasympathetic function. When we looked at that alone in relationship to depression, there was no significant relationship or correlation. Same with pre-ejection period or sympathetic activation. Just alone, no notable relationships. However, when we looked at the interaction of the two branches, what we found was that overall, our individuals with autism had higher reported affective symptoms as we would expect. And it was only those who had both low sympathetic activation or activity in this kind of barred graph here and high RSA or high parasympathetic. Those were the only individuals that showed lower affective symptoms relative to anyone else within the autistic group who all maintained higher depressive symptoms regardless of physiological profile. And so what this really told us was that, again, not only are we looking at these, interrelated behaviors and profiles, but also thinking about the idea that there are two branches here. And although focus has largely been on the parasympathetic system, less is known about sympathetic. But clearly, especially when we think about potential clinical implications, we do need to take both sides of the story into consideration, because it may not be one in isolation, but the interaction of the two that's really important to get a full picture of these physiological profiles and how they relate to behavior or psychological functioning. One of the other questions that we raise, both in terms of future directions, but also in terms of key takeaways, is the utility of heart rate variability as a biomarker in autism. The goal or the hope has been that it could be used as a biomarker, and in many ways, very well may be. However, we emphasize the fact that you also need to think about this from a transdiagnostic approach, and thinking about not just categorical determination of autism or not, but even within autism subgroups and those with more depressive or anxiety symptoms. Is there something unique about their response profiles that's important for us to identify? And so really focusing on these individual differences and traits across a number of domains, including social and psychological and even physical. And then this would also, in turn, potentially help with identifying possible areas of intervention. So if we can identify specific profiles that are indicative of an increased risk for, say, developing a depression diagnosis or anxiety, is there something that we can do earlier on to intervene to help, alleviate some of those concerns? And with that, just highlighting our primary takeaways. So while there are there is evidence to support differences in autonomic function in autistic individuals, these individual characteristics do matter. And that's the biggest thing that we've taken away from our research up until this point is when we think about the influence of BMI, medication, social behavior, internalizing profiles. These are all really important characteristics to keep in mind when we're trying to define how physiology or autonomic function is different, if at all, in autism. And then again, these implications for psychological functioning, especially when we think about internalizing conditions and that relationship between our bodily response and then our brain, our cognitive interpretation. And so how might this, also impact, things like anxiety and depression? And then again, lastly, there's more work that needs to be done to better understand if heart rate variability or the autonomic system as a whole could be utilized as an ASD specific biomarker, or is it more transdiagnostic in focusing on specific characteristic profiles that might be more informative in terms of identifying areas for intervention or identifying particular profiles that might benefit most from certain types of intervention. So with that, I just want to importantly acknowledge the Sense Lab and especially Dr. Blythe Corbett at Vanderbilt. I started off as her mentee and have continued on now into a faculty role, but I still collaborate extensively with Dr. Corbett in the Sense Lab. And so I certainly want to acknowledge her role in all of our research, as well as the role of all of the Sense Lab team members and collaborators and our undergraduate students who have put in so much time and energy into this research. And of course, always want to thank the children and the families who participate in our research. Without them, none of this would have been possible. And so we are incredibly grateful for them devoting their time and energy to participating in our studies. And so with that, I can open it up for questions.
Okay, great. Thank you so much. There were many, many questions. So I'll start to read through some of those for you. Okay.
Sure.
So some of these are pretty complicated questions. So I
okay.
start off by just acknowledging some things. Some people were asking questions about sensory issues related to all of this and that how that ties into that. And I know this isn't the big focus of what you were talking about today, but we do on our website have other webinars about that where we've really we dive into some of those sensory issues with different researchers. So I'd encourage people to go and watch those if they haven't seen those in the past. And then we also have Blythe Corbett has also participated and presented webinars for us. So if people are interested in following up with that as well, it'd be great to check that out. So starting off here, I'm going to start with this question about the influence of BMI. So you're talking about the influence of BMI. So is that the psychological impact? Is it just the physical impact? Is it everything? What is the suspicion here? I don't know what you do know or don't know, but that question more than once.
That's actually a really interesting question. In our as we've thought about it up until this point, we've really thought about it from a physical standpoint. And the way that BMI or body weight can impact the cardiovascular system as a whole. And so, we do know that there are health risks associated with increased body fat, such as, heart attack, stroke. And these are also, these cardiovascular diseases are also associated with differences in autonomic function. And so we can actually use the balance of the parasympathetic and the sympathetic branches to potentially, predict what might be most at risk for heart attack. So we've always thought about it in that context, in the physical context of its the overall impact of the cardiovascular system. However, thinking about it from a psychological perspective is interesting. I don't know exactly what might have been the intention, in thinking about that, but if you think about even just the unfortunate piece that there is a lot of a social evaluation component to our bodies, and especially during the adolescent period, but also just in general. And whether or not comparing ourselves to others also helps, also contributes to some of that. Oh, I am so sorry. There's a cat.
Oh no, it's great. We all have that.
He made an appearance. That was So, but yeah, no, the psychological piece is interesting to think about. But we have largely been considering it from a more of a physical role.
Got it. Okay. Well, thank you. And
Mhm.
certainly it's a complicated topic and probably a lot of
Yes.
research will be happening in that area. People were asking about where the playback's going to be. I just want to tell everyone it's at autismwebinars.org, or you can just go to autism.org and click webinars and you'll be able to find it. We'll process the playback as soon as the webinar is over, so it should be up later today. So if you want to share it or review parts of it, it will be out there. And yeah, someone just acknowledged your very sweet kitty.
No, thank you. His name is Stuart. Yes, he's a cutie pie, but he also gets into trouble like jumping onto Zoom meetings. So
Yeah. Yeah. Well, I think Zoom actually emits some kind of sound because I have a cat that is always really interested in that sound, whatever it is.
Mhm.
It's something I can't
Yep.
hear. Okay, so
Yep.
this is another question. They're asking, is there any research of interest on vagus nerve stimulation for autism?
Mm.
So do you know anything about this, or is this something they should go and research or speak about with their physician?
So you can ask your physician, you can speak about that. I don't know how, in terms of the medical field, how much is really known at this point. I can direct you to uh, one of the theories that I cited, the polyvagal theory. There is an entire institute looking at vagal nerve stimulation in relation to the polyvagal theory. So if you just kind of search polyvagal and Stephen Porges, there's more information. But as far as the research into it and clinical utility or safety, I'm not well versed in that at this point in time.
So this is a question about diagnosing depression in autism. So in the
Mhm.
context of the studies that you talked about, or other studies that you're aware of, what are the different instruments that they use to assess depression and autism?
Sure. Yeah, no, that's a really great question, and is something that is driving some of our research as well. Is that up until this point, most of what we think about when we think about diagnosing depression comes from clinical interviews, or as both parent and self-report. But in autism, that could be that could be challenging, especially when we think about if an individual is unable to truly verbally, express how they're feeling, because so much of that diagnosis is based off of how somebody, what somebody says they're feeling, or what they're thinking. And so that would be, kind of a perfect world down the road kind of avenue for our research is to use something more objective, like a physiological measure, such as heart rate variability, actually as a biomarker for something like depression as well, to help with that diagnostic determination. But yeah, up until this point, like it's very largely based off of clinical interview with either a psychologist or an MD.
So I don't know if you can address this or not. When you talk about
Mhm.
medications that can impact some of these, that are co-occurring at the same time when you're looking at considering all these factors, were there any specific medications that were mentioned, like risperidone or anything else?
Oh,
Yeah.
nothing specific. We have not gotten into that level of detail, but just simply, kind of broad category psychotropic medications at this point. But nothing specifically. No.
Someone's asking about using copies of your slides. They do say for internal use only on that. So do you want to comment at all on that?
I would say if they could email me, that would be great. I don't know if there's a way I can provide my email address, and that might be the best way to do it.
I think
I don't know.
people can email me and I would
Okay.
be happy to
Yes,
contact you.
that would be perfect.
I'll
That
forward
would
that.
be, yes, absolutely. Yeah.
Okay. Okay. So a bit of this person's asking about specific research of brain regions. So are you aware, and I
Okay.
don't want to be on the spot, you may not know,
Mhm.
are you aware of specific brain regions such as Broca's or Wernicke's area that show altered activity in people with autism with speech delays? And if this is outside of your knowledge base, that's totally fine.
That it's an interesting question, but yeah, those areas in particular and speech and all that, that's outside of my area of expertise. But it is an interesting question.
So you may have addressed this, so I apologize if
Mhm.
I'm asking you. I was multitasking. So do the results apply equally for female and male autistic people?
Uh, Great question. Thank you. As of right now, yes, but there is a major disclaimer that in all of our research that I reported today, our females were very much underrepresented in our sample. It was, it certainly wasn't a 50-50 split. It was, I think, a 3-to-1 or 4-to-1 ratio. And so in some cases, we had as few as like 20 to 25 females. I think Dr. Corbett touches on this in her talk from a couple of weeks ago, but we do, the Sense Lab as a whole has a larger study right now looking specifically at females. And that is part of the goal as well, is to better tie in the physiological piece as well, and whether there are sex differences. Because we do believe that there are going to be sex-based differences. We just haven't quite had the power yet to be able to look at that. So to be continued.
All right. So in this work, did you also look at all at self-injury, or was that separated out from what you were working on?
Yeah, so we do have that information somewhat. It's not a when we kind of accounted for it, it wasn't a great measure. But we haven't really specifically looked into that. I do know there are others in the field who are very much looking at autonomic response and self-injury behaviors or aggression. But that's not our particular area. But there are others in the field who are definitely looking into
People are still asking for your email address. I put my email address in the
that.
chat. If people would want to send questions to me, I'll be sure to forward those
Yeah, that'd
on.
be lovely.
Okay. So people are asking a little bit more about just if autistic people are experiencing depression, different
Mhm.
symptoms that were addressed. I know this study was not about treating necessarily, but
Correct.
right. But if people feel like they are supporting a loved one or themselves are experiencing those things, where would you refer someone, or what would you suggest their next step should be?
I definitely suggest talking to your physician, primary care, or if you have a relationship with a psychologist or some like a psychiatrist, certainly at a medical professional, a trusted medical professional, to reach out and to get support. And they can certainly direct you more towards specific resources for your area.
Right. So questions, if you have questions about cognitive behavioral therapies, or there's sorts of things, or if you want counseling or different things
Yeah.
like positive support in
Mhm.
the community,
Yeah.
often those things are community based. So asking
Mhm.
people in your own community can be the most impactful way
Yeah.
to find the
Yeah.
appropriate resources there.
Yeah.
And in the
Definitely.
post-COVID world, there's a lot of online resources
Yes.
as well. And for people who
Mhm.
experience anxiety or depression, sometimes that's a safer route.
Mhm.
So something to consider
Absolutely.
too. Okay,
Absolutely.
this person's asking more about weight gain.
Okay.
Was there a correlation between like as weight increased, were
Mm.
symptoms worse, or was there any difference? Is there a correlation between the amount of like the BMI,
Sure.
higher BMI and more impact?
So, yeah, that's a great question. So, we did look at that in our most recent study. We looked at change in BMI because we measured over 4 years, an individual's change from year one to year four. That was surprisingly to us not related to the physiology at all, the parasympathetic or sympathetic. It was only just like average BMI, just overall, especially at year one. The year one time point was really the most critical. That was related to those physiological profiles. Like I said, that was surprising to us because there is, some research in, adult neurotypical populations that do suggest that weight change over time could either have a positive or a negative effect. So it's something that we do want to look into a lot more because we want to better understand how that, piece might be contributing and as we think about, try to think about like causal relationships or things like that. And, the directionality of that relationship between physiology and physical health. So we haven't seen anything yet, which is surprising, but we certainly are planning to continue to look into that further.
Okay, so this has some, there's some acronyms here, and I'm not sure entirely
Okay.
sure what they're getting at.
We'll test my acronym.
Right, you mentioned utility of HRV as a
Yeah.
biomarker needs more research.
Mhm.
Devices that measure HRV are becoming more commonplace.
Mhm.
Could there be any practical application of that for autistic people as they consider regulation? Or is that something that we can't speculate about?
So I don't want to speculate too much on that, but I do want to again say that there are people who are looking into that. I don't know enough about what they do, and I'm not them. They are the experts in that area, but there are people who are diving into that a little bit more. But unfortunately, I don't know enough to really be able to speculate, other than to say that it is on people's radars, and it's something that we're trying to better understand if there's something that can be utilized there.
Okay. Have you, in any of this work, ever looked at biofeedback or mindfulness-based interventions? Has that ever been part of the work, or is there anywhere you'd refer people for that?
So I don't have any great recommendations for where people can go for that. It is a very important and very valid question. But it's not something that we have gotten into at this point in time. But I can certainly understand the curiosity around that. And but yeah, unfortunately, I don't have any specific recommendations on where one might go for reading more into that.
This person's asking, and you addressed this somewhat, but I think they're asking for clarification. So,
Sure.
In the work that you did on depression,
Yeah.
was depression more common in high functioning autism and more linked to social isolation, sensory overload, or executive functioning challenges?
So yes to the first part of the question. Yes, the uh, autistic individuals had higher depressive symptoms. We did not look at clinical diagnosis of depression. We just looked at depressive symptoms as reported by the individual or the parent. And that was higher in our autistic youth, but we did not look at executive attention or sensory processing, anything like that. I should also mention, it's really important to mention, I should have done it earlier. The research up until this point has been largely in individuals with autism without intellectual disability. And they do have to have at least some spoken language as part of the overall study requirements for the interaction and things like that. So that is a limitation of our work so far, is we're not capturing, this, the full spectrum. And so we can't say how, some of these relationships might differ when we think about, different symptom profiles.
So this person's asking, what's next? What's the next phase in this kind of work? What do you study as you start to think about these questions in a new way?
Yeah, so right now, the one of our kind of more intriguing projects is looking at the something in terms of physiological synchrony. So up until this point, we've looked a lot at the individual's physiological response or stress response during these interactions. But there is a key component in terms of how two people's both behavior and their body, their physiological function, actually work together when you're having an interaction. And it's that kind of synchrony that can actually really help support, strong positive interactions. And so we are very interested in the extent to which there's actually any, synchrony differences, or to what extent that might actually be playing a role in some of what we're seeing with these social difficulties in relationship to autonomic function. So that's definitely a big piece of what we're looking at. But I also think too, very much focusing on the trait-based idea and looking beyond thinking about it from a categorical perspective. But we have a very rich data set of a large number of individuals now. And so we want to take advantage of that by looking at a number of different individual characteristics. And, if we can get more into kind of subtyping profile type findings rather than just autism versus not.
Okay, and I know this wasn't something that was really a focus of this particular talk, but I had a couple questions about sleep and how that mixes into all of this, and that follows up. We had a talk on sleep last week, so I think we have some folks who that's a real focus for them. Were there any findings in this work that related specifically to that, or is there a future study in that area related to this?
So we don't have anything yet. The thing about with sleep that's tricky is that we aren't monitoring over 24 hours. Like all of the physiological measurements are made in the lab. And so it's difficult to kind of associate that with sleep patterns. Dr. Beth Mallow, who did give the talk last week, she does have one publication with some collaborators that they actually looked at heart rate during sleep. But up until this point, we haven't really gotten into that area, largely because so much of what we're doing is measured in the lab, and so we're not collecting actually when somebody is sleeping.